Hello everyone, hope you had a good Easter weekend. Let's go ahead and jump right into lipid metabolism. Okay, so how does our body get energy? Up to now we've really focused a whole bunch on glucose, but as far as I know, none of us are eating just straight glucose for every meal. At least I'm not. I've had a lot of meals. So today we'll be talking about our other main source of energy, dietary lipids, or fat. So if you see on a nutrition facts thing on the side of your cereal box or something, you'll see fat, cholesterol, sodium, carbohydrates, and protein. And you'll see that fat right there at the top has the most calories per gram. So right here. So let's talk about how those calories get into your body, how your body makes use of fat for metabolism. Today we'll have 4 learning objectives with a couple of sub-learning objectives. First, we'll describe the process by which fatty acids enter our cells. We'll calculate ATP equivalents gained by full metabolism of fatty acids, similar to what we already know how to do for glucose. We'll extrapolate beta-oxidation of fatty acids to other atypical fatty acids. And we'll integrate lipid and carbohydrate metabolism together to see what our body does when we eat and when we're hungry. And the 2 sub-objectives, we're going to identify ketone bodies and identify malonyl-CoA as sort of a regulator for fatty acid oxidation. And those fold into objective number 4. All right, dietary lipids. How do you get lipids inside of your body? Well, there's 2 sources. There's exogenous, from outside the body. These are derived from dietary fats. So these exogenous lipids are lipids that you eat. Endogenous lipids, or lipids from inside, is the biosynthesis of fatty acids from excess dietary carbohydrates and proteins. So these are lipids that you make yourself. Now, we're just going to be talking about exogenous lipids today. Endogenous lipid biosynthesis is kind of out of the scope of this class, but it is in your book. There's actually a pretty large chunk of it in your book if you want to read it. But today we're just going to be talking about exogenous dietary lipids. Okay, so when you eat something that contains lipids, something that contains fat, where does it go? So when you eat it, something called chyme is produced in the stomach from ingested food. And this mixes around with bile in your small intestine. And this mixing causes emulsification. And this makes small lipid droplets, or micelles. You might remember back to chapter 3. Lipids can combine to form micelles. And this is where it becomes relevant. So your pancreas secretes enzymes and proteins into the duodenum, and these help digest your food. And fatty acids from these can be passively transported across the cell membrane. So here's sort of a schematic. You should be able to find this in your handout as well. So starting from your stomach, bile and chyme enter your small intestine. They churn around, form these micelles, and your pancreas secretes these enzymes, which is lipase and colipase, to break these down even further, where they're then absorbed through your intestinal epithelia into your body. So once they're inside the cell, these lipids, probably in the form of triacylglycerides and cholesterol, they're reformed. And these reformed little chunks of fat, these lipid particles, are called chylomicrons. So a good way to remember this is maybe chyme, chylo, and then microns are small. So they're small little particles that consist of, they're little tiny fat chunks, so to speak, microscopic fat chunks. They got triacylglycerides, phospholipids, cholesterol, and they're sort of bound together with some protein. So these chylomicrons, once they leave your intestinal epithelia, your intestine, enter the lymphatic system and eventually make it into your bloodstream. Now once they're in your bloodstream, those triacylglycerides in the chylomicrons get hydrolyzed back into fatty acids and glycerol and are uptaken into your adipocytes or your fat cells. So in the fed state, when you've eaten, the fatty acid concentration is pretty low in your blood. But when you're hungry, this can rise 100-fold. Why is that? You know, think a little while. Write something down in that box in your handout, and sort of conceptually think of why this concentration might change. And pause the video and come back when you're ready. Alright, so this is what happens, then I'll sort of explain why it happens. So, when you're hungry, in the fasting state, your blood glucose will fall, and epinephrine will be released from your adrenal glands, and it will bind to receptors on your adipocytes, on your fat cells. And that'll cause the hydrolysis of those stored triacylglycerides, triglycerides, and it will release free fatty acids into your body. Now, as for why this happens, why those concentrations rise 100-fold, this really gets back to our uniting idea of whenever your body needs something, it will do what it takes to get it. If your body needs energy, it will have mechanisms to release fatty acids into your blood when you need it. So this is the mechanics of how it happens, by releasing epinephrine to cause your fat cells to release fatty acids. But why it happens is because your body needs energy. So that's sort of a unifying thing. If your body needs glucose, gluconeogenesis, it has a way to do that. If your body wants to store glucose, it can make glycogen. It has a way to do that. If your body needs energy now, it has a way to release fatty acids. So how do we extract energy from these fatty acids, from lipids? It's through a process called fatty acid oxidation. And it actually oxidizes to acetyl-CoA. So there are 3 steps: activation, transport, and beta-oxidation. So oxidation consists of 3 steps, or fatty acid oxidation consists of 3 steps. First, we got to activate that fatty acid. So this activation is the conversion of fatty acids to acyl coenzyme A. So there's CoA again. And now we're attaching a fatty acid to it. And this occurs in the cytosol of cells. Then we need to transport the fatty acid into the mitochondria. Looks sort of similar to the Krebs cycle, I suppose, or the oxidative phosphorylation. Finally, we need to actually oxidize the fatty acid to extract the energy out of it. So this is the conversion of fatty acid, fatty acyl-CoA, to acetyl-CoA, which then does go into the Krebs cycle. And this occurs within the mitochondria. Alright, so let's dig down into this a little bit. So activation. This converts a free fatty acid to a CoA ester. And I've put the equation below. So a fatty acid, designated here as RCOOH, plus CoA, CoA ends in a thiol, so this is just the standard CoA that we know and love, plus 1 unit of ATP, react to form this fatty acid CoA ester that I've drawn right here. The fatty acid clicks on to the sulfur, the thiol group in CoA, plus 1 equivalent of AMP and an inorganic phosphorus. So you might note that activating this fatty acid into a CoA ester takes some energy. ATP goes all the way down to AMP. So this reaction is catalyzed by fatty acyl-CoA synthase. That's the enzyme that performs this reaction. And it occurs in the cytosol of cells. It's metabolically irreversible. And it takes 2 ATP to activate. ATP goes all the way down to AMP. So similar to glycolysis, if you want to draw a link back to glycolysis, it takes a little bit of energy investment to get your, in glycolysis, to get your glucose inside the cell and ready for glycolysis. Same sort of thing in fatty acid oxidation. You have to invest a little bit of energy to activate that fatty acid and oxidize it. Okay, step 2. This is the transport of your activated fatty acid into the mitochondria via something called the carnitine shuttle. It was originally discovered in muscle cells, so carnitine shuttle. The fatty acyl-CoA, that activated fatty acid, is converted to a carnitine ester by Cat1. So just another enzyme. And the carnitine ester would be shown in this figure right here. And so the mitochondrial transport protein catalyzes the exchange of the activated or the acyl carnitine in the cytosol for a mitochondrial carnitine. So that's a little bit of a word salad there. So let's draw out what's going on. This transport protein, this pink oval right here, switches a fatty acid attached to carnitine outside the mitochondria with just a free carnitine that's inside the mitochondria. So it takes this into the mitochondria, simultaneously takes a free carnitine, takes it out of the mitochondria. So it swaps them. So once your fatty acid carnitine is inside your mitochondria, another enzyme, Cat2, converts your fatty acyl carnitine to fatty acyl CoA once again. You basically regain that activated fatty acid from step 1, and that carnitine gets recycled back out to the cytosol. It's a pretty crafty little trick there. Recycles your carnitine, saves you a bunch of energy. You don't have to keep making new carnitine. So if you didn't get the picture from the previous diagram, we can take a little closer look at this picture right here. So step 1, we've got to activate that fatty acid by attaching it to CoA. So we got acyl CoA. And then for transport, we swap that CoA with the carnitine, put the carnitine inside the mitochondria, and then swap the carnitine back for a CoA. Basically, the carnitine is just acting as a transport helper. It's not part of the oxidation itself. It's just transporting that fatty acid inside the mitochondria. So now we're going to talk about the third step, which is beta-oxidation. This is where we're actually going to be extracting energy from the fatty acid. So how does it work? Each round of beta-oxidation removes 2 carbons at a time from the fatty acid in question, and it converts them to acetyl-CoA. And that resultant acetyl-CoA can enter the Krebs cycle. And it also generates 1 unit of NADH and 1 QH2. So, like I sort of mentioned already, beta-oxidation is a cycle. Each time you go through the cycle, you remove 2 carbons from your fatty acid. And actually, it looks similar to the Krebs cycle. It's only 4 steps long. And these steps are repeated as needed. So if you chop 2 carbons off your fatty acid and you've still got 12 carbons left, you can keep chopping. You can go through the cycle over and over again until you're done, until there's no more carbons left. So here's a picture of it. We're not going to go through step by step and memorize each individual step that occurs. But I'm going to talk you through it on this slide just so you know sort of what's going on, where the carbons are moving. So let's start off with our activated acyl-CoA right here. We've got the fatty acid portion, and here's the CoA portion. So it enters the cycle, and it generates 1 FADH2, which we know will be converted to QH2 in oxidative phosphorylation. And you don't have to know exactly what each reaction does, but we'll talk about the parallels to the Krebs cycle here in a second. So it goes through the cycle. It's already generated 1 FADH2. Here it generates 1 unit of NADH. And here, these 2 carbons highlighted in red get kicked off to make acetyl-CoA. So we know that that's going to go into the Krebs cycle. And what we get is the same fatty acid minus 2 carbons. These 2 carbons are gone. But now we've still got a fatty acid. It's just a little bit shorter. So we still got that fatty acid, so let's just keep on chewing. And it'll release one more FADH2, one more NADH, and then one more acetyl-CoA, and so on, until the fatty acid is completely degraded. Okay, so here's a little image just to show sort of the similarities between beta-oxidation and the Krebs cycle. You don't need to, once again, you don't need to memorize this at all. I just want to sort of show how these are so similar. Yeah, just take a second, look at it. We're looking at these 2 carbons on the left for beta-oxidation. They're highlighted in yellow. And on the right for the Krebs cycle, they're comparing the 2 carbons highlighted in yellow. So it's sort of logically the same sort of reaction strategy, and it generates similar amounts of products. So how many times does beta-oxidation need to take place for any given fatty acid? I've been saying how beta-oxidation occurs until the acid is completely chewed up, until there's no more carbons left to chew. So the golden rule for this is the number of carbons in your fatty acid divided by 2 minus 1. So let's look at this example. So here we have capric acid. You can see that for capric acid there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 carbons. So 10 divided by 2 is 5. Minus 1 is 4. So what I'm saying is that for capric acid, which has 10 carbons, it will require 4 rounds of beta-oxidation to completely chew it up. So here I've said 4 chops. So the first round of beta-oxidation will chop here, and then it'll enter the cycle again, chop here, then again, chop here, and then finally chop here. We don't have to chop here because, well, for the last 4 carbon fatty acid, if you chop that into 2, you already get a 2 carbon species. So the final chop actually makes 2 equivalents of acetyl-CoA. So that's kind of nice. The last step, you sort of get that one for free. Let's try doing this ourselves. So 1 round of beta oxidation makes 1 QH2, 1 NADH, and 1 acetyl-CoA. And the final round, the last chop of beta oxidation, makes 1 QH2, 1 NADH, and 2 molecules of acetyl-CoA, because we don't have to chop it again if we're at that last one. So for a standard round, the theoretical yield of 1 beta oxidation is 17 ATP. We've got 2 for QH2, 3 for NADH, and 12 for acetyl-CoA. Because as we already know, acetyl-CoA will go and enter the Krebs cycle, and that's 12 ATP. Though of course, the very first time you put that fatty acid into the mitochondria, that costs you 2 ATP. You don't have to do this for each round of beta oxidation, just the first time you put that fatty acid into the mitochondria. So, try this for yourself. How many ATP are generated from complete metabolism of capric acid, which is 10 carbons? So, take a second here, pause the video, and try the math for yourself. When you think you're ready, go ahead and unpause it, and we'll walk through it together. All right, so here is what I am thinking. Oops. So it costs 2 ATP to get your fatty acid inside mitochondria. So let's just start with a -2 right off the bat. Next, capric acid is going to need 4 chops. Remember, the number of chops we need is number of carbons, which is here 10, divided by 2 minus 1, because the last chop you get 1 extra acetyl-CoA for free. So if we have 4 chops, that means we get 4 times the products of each chop, the products of each round of beta oxidation, which, just to remind you, 1 NADH, 1 QH2, and 1 acetyl-CoA. So, well, 1 acetyl-CoA plus 1 QH2 plus 1 NADH is 17. And if we have 4 chops, that's 4 times 17, which is 68 ATP. So remember, each round of beta oxidation makes 17 ATP, theoretically. So if we have 4 rounds, 4 times 17 is 68. But remember, that final chop gives you a bonus acetyl-CoA, so a bonus 12 ATP. Okay, so now we're done. We've gotten our fatty acid inside the mitochondria. We've completely chopped it up using beta oxidation, and we've even included that final bonus acetyl-CoA from the final chop. So our total is 12 from that bonus acetyl-CoA, 68 from beta oxidation, minus 2 for the cost of getting it inside the mitochondria, for a grand total of 78 ATP. So if you wanted to compare this to glycolysis, we get a whole heck of a lot more ATP from metabolizing 1 fatty acid than we do from metabolizing 1 unit of glucose. And this isn't even a particularly large fatty acid. This is a 10 carbon fatty acid. Glucose has 6 carbons. So glucose gives you 38 ATP theoretically. This can give you 78 ATP theoretically. So lipids can be metabolized by your body for a lot more energy, molecule for molecule, than straight glucose can. Okay, so this is nice. It's a little bit complex, but now we can do the math. Great. So it makes sense for saturated fatty acids with an even number of carbons. But, you guys have been through organic chemistry. There's all kinds of fatty acids. We've talked about saturated and unsaturated fatty acids, even-numbered fatty acids, odd-numbered fatty acids. There's all sorts of varieties. What does our body do with other kinds? So basically, for all of these other varieties, it uses ancillary enzymes, ancillary reactions. So you can think of these as extra enzymes. And they're needed to completely oxidize unsaturated, very long, or other atypical fatty acids, odd-numbered fatty acids. So for example, unsaturated fatty acids. So these are fatty acids that contain a double bond in the carbon chain. And these are pretty abundant. We already know that these are used in, for example, cell walls of animal cells. So these can be oxidized similar to saturated fatty acids, but they require a tiny modification. So here's an example. And for what it's worth, you don't need to memorize exactly how all of these go. You just need to be aware that atypical, which is to say, if atypical fatty acids are metabolized similar to typical saturated even number fatty acids, but they require a couple extra steps. So here's an example just for your own personal edification. This is oleate, or I guess oleate-CoA would be the better way to say it. So oleate-CoA. It's a long chain fatty acid, and it's got an site of unsaturation right here. So how this gets metabolized is it'll get imported into the mitochondria and undergo 3 rounds of beta oxidation until we're right up against that unsaturation. So then it has to undergo an ancillary enzyme reaction, here an isomerase, to flip it around and get that ready for metabolism, basically. So now that it's right next to the carbonyl, it's ready to get metabolized. And it'll probably get reduced first, and then it'll get completely oxidized. So once again, do you need to memorize how this happens? No. You just need to be aware that if you've got an atypical fatty acid, it's going to require some extra enzymes, ancillary enzymes. So an isomerase will then clean it up, and once it gets cleaned up, beta-oxidation can occur. Same thing for odd-numbered fatty acids. These require additional ancillary reactions. So very short fatty acids, these actually bypass activation altogether. They're just so short, they get activated within the mitochondria. So they can pass in through porins, for example. They don't require the carnitine shuttle. You can pass, so they bypass step 1 and step 2, basically. And very long fatty acids, they're actually partially oxidized elsewhere in something called a peroxisome. And unfortunately, the oxidation of these very long fatty acids are not energy linked. So you don't get a lot of energy from very, very, very long fatty acids. For what it's worth, your body doesn't really use these as food. So very, very long fatty acids are typically foreign antigens or portions of foreign antigens. So your body's not set up to use them as food. And that's okay, because we don't eat a lot of them. All right, so now we've talked about how to metabolize saturated, even-numbered fatty acids. And we've also extended that idea to atypical fatty acids, which is to say unsaturated fatty acids, or maybe odd-numbered, or very long or very short fatty acids. So now let's focus in on ketone body formation, or ketosis. If you work out a lot and you're familiar with the concept of ketosis, this is where this fits into our metabolism. So if we're talking about ketone bodies, most of the time biologically what we're talking about are things related to acetone. So acetoacetate, beta-hydroxybutyrate, and acetone itself. They're known as ketone bodies. And this is sort of a dumb name. They're not bodies. You might as well just call them ketones, for what it's worth. They're called ketone bodies because they're ketones. So they're water-soluble ketones. So these ketone bodies are produced in the liver. And they're produced during a spillover pathway that occurs during excess acetyl-CoA production. So if you make acetyl-CoA during the Krebs cycle or fatty acid oxidation, and you make a whole bunch because you're working out really hard, you need a lot of energy, you'll produce a lot of ketone bodies. So yeah, usually when this happens in your body, it's due to excess fatty acid oxidation. So if you need energy and your body is outputting a lot of free fatty acids, so your free fatty acids in your blood are increasing, they'll also in parallel have a rise in the number of ketone bodies. This makes sense. This is easy to grab onto because if we're outputting a lot of free fatty acids, well, we're going to use them. And we just learned that using these free fatty acids, oxidizing them quickly, will lead to a rise in ketone bodies. So here's an example. Let's just focus on the top part here in red. So 2 units of acetyl-CoA can react to form aceto-acetyl-CoA, shown here. So we lose 1 CoA, and you get aceto-acetyl-CoA. And you can see this is a ketone, so it's a ketone body. And these things can react further to make different species, different ketone species, different ketone bodies. So aceto-acetyl-CoA, just for example, is eventually reconverted into 2 molecules of acetyl-CoA, and then that enters the Krebs cycle. So what's the relevance of this to our metabolism? So many tissues can use these ketone bodies for energy, these sort of side products of fatty acid metabolism for energy. So the number one example is your brain. So your brain uses ketone bodies in severe starvation. It prefers glucose, but if it can't get glucose, it'll make do with ketone bodies. Though I recommend glucose because if you're getting to the point where your brain is eating ketone bodies, you're probably not feeling too well. So keep the ketosis to other organs, please. So yeah, unlike fatty acids, ketone bodies can cross the blood-brain barrier. So they're used for fuel by the brain if they can't get any glucose. Great. So now we've got under our belt, we've got glycolysis, a lot of reactions downstream of glycolysis. We've got lipid metabolism, and we've got glycogen. We've got all sorts of things that are related to how our body intakes and stores energy and releases that energy when it wants it to. So let's tie this all together. Let's finish up today by talking about lipid and carbohydrate metabolism in the fed and fasted states. So in the fed state, when you've eaten, dietary lipids that you eat are packaged in chylomicrons, as we know, and eventually it makes it to fat cells. Whereas carbohydrates, they're stored as glycogen in the liver and the muscle until the maximum glycogen concentration is reached. And then they're converted to fat. Once again, we're not talking about making about endogenous lipids, but just know that they do get converted to fat. And they're packaged in something that's called VLDL, very low density lipoprotein. That's also stored in fat cells. So if you've eaten, lipids get stored as chylomicrons, carbohydrates get stored as glycogen until you've got enough glycogen and then stored as fat. So as you know, the major hormone involved is insulin. So you know that insulin's involved in carbohydrate energy regulation, same thing for lipid. So insulin stimulates fatty acid synthesis in the liver and fat cells, and it favors triglyceride formation. It favors storing those fatty acids in your fat cells for later. So insulin favors storing glucose as glycogen and fatty acids as triglycerides. So it's an anabolic hormone, favors biosynthesis of large molecules. How about when you're hungry? What if you haven't eaten for a while? Now your body's going to need that energy that it saved up from lunchtime or whenever. So fatty acids, free fatty acids, are going to be produced by breaking down those triglycerides in fat cells. Insulin caused these to be stored as triglycerides. Now epinephrine is going to stimulate your fat cells to release those as free fatty acids. So once those are in your blood, your liver uptakes those fatty acids and oxidizes them to CO2, or if you really need a lot of energy, along the way they'll get partially oxidized to ketone bodies, as we mentioned. So for fat cells, you've got insulin, which causes uptake, and epinephrine, which causes release of fatty acids. But we also have another regulator of fatty acid oxidation, malonyl-CoA. So here's CoA again, that we know and love. And malonyl-CoA regulates fatty acid oxidation by inhibiting it. So it inhibits the metabolism of fatty acids. And it's the precursor to fatty acids in liver and fat tissue. So here's sort of a wrap-up of it. Only fat cells provide fatty acids for other tissues. So you know how your glycogen in your liver provides sugar to the rest of your body? Only fat cells provide fatty acids to the rest of your body. So it's stimulated by epinephrine and it's inhibited by insulin. Okay, so fat cells synthesize, store, and release fat when they're told by insulin and epinephrine. Muscle cells, they use fat and ketone bodies for energy. Liver synthesizes fat, secretes VLDL, and if you're hungry, metabolizes fatty acids and makes ketone bodies. So that's sort of how these different cell types interact with each other. Okay, so we know what your body wants when it's fed and when it's hungry. How about the transition between the fed state and the fasted state? So, of course, this is going to be characterized by storage pathways. So, glycogen for sugar, triglycerides for fatty acids. So, when you eat, insulin becomes elevated. Malonyl-CoA levels are high. So, remember that inhibits fatty acid oxidation. And when you're hungry, glucagon increases. Liver glycogen begins to break down to provide blood glucose, and fatty acid synthesis is stopped by inhibiting acetyl-CoA carboxylase. So it doesn't store fatty acids. It uses them for energy. So along with this, malonyl-CoA levels drop when you're hungry. So malonyl-CoA inhibits fatty acid oxidation. If there's no more malonyl-CoA, oxidation will no longer be inhibited. So now you can use those fatty acids for energy. So this increases ketone body formation. And so epinephrine in your blood will become elevated, as we know, to stimulate your fat cells to release free fatty acids. So basically the storyline here is if your body needs energy, it has all these pathways to get that energy. Breakdown of liver glycogen, release of fatty acids from your fat cells, stopping inhibition of fatty acid metabolism. So make sure to pull all these levers to make sure you get your energy when you need it. So, well, why is this so important? I mean, inherently, you sort of know why it's important, because if you didn't have all these things, you would die. You wouldn't get that ATP that you need so very much. So the maintenance of blood glucose is important. So for example, we already talked about it, your brain uses glucose primarily. If you don't have any blood glucose, you can use those ketone bodies, but gluconeogenesis is going to be really important to make that new glucose that your brain needs. Your brain can't use fatty acids. The rest of your body can. If you can't get glucose, you can't make new glucose through gluconeogenesis, and you don't have any ketone bodies. You're going to start breaking down proteins. You're going to get your energy from amino acid breakdown. And that's sort of like the metabolite of last resort for your brain and other organs. So, once you've gotten that far, you should really think about, you should eat something. Okay, one last thing. So this is during exercise. So I think this is sort of interesting. It's not actually on your handout. It's sort of a fun fact type of slide. So during exercise, you're working really hard. Blood flow is shunted away from most tissues, except for your muscles, which are working out, and your brain, which is, your brain. It's the exception for everything. So suppose you're lifting weights, you're doing anaerobic exercise. So your muscle is locally breaking down stored glycogen. So, activation of glycogen phosphorylase, no effect on lipid metabolism. Anaerobic exercise, you're just using your local glycogen. Pretty neat. How about aerobic exercise? Suppose you go and jog 50 miles or something, whatever you do in the morning to wake yourself up. So if you're doing aerobic exercise, epinephrine will lead to the release of fatty acids from your fat cells, which will then travel to your muscles and become oxidized, in addition to that breakdown of stored glycogen. So basically what this says is aerobic exercise has the ability to burn fatty acids, whereas anaerobic exercise just uses local stores of energy. So aerobic uses both, and anaerobic just uses local. So I think that's pretty interesting. Two different methods to get your muscles the energy that it needs to succeed. Okay, let's summarize. So after you eat, fatty acids enter your bloodstream as chylomicrons, and then they get stored eventually as triglycerides in your fat cells. And the usage of fatty acids as energy is called beta-oxidation. And it's kind of similar to the Krebs cycle, and it produces 17 ATP equivalents per round, except for that last round, that last chop, which makes an extra 12 ATP in the form of acetyl-CoA. Beta-oxidation of atypical fatty acids requires ancillary enzymes, extra enzymes, and fatty acid metabolism is regulated similarly to carbohydrate metabolism. And I don't know if I really made this clear enough, but it's regulated similarly to carbohydrate metabolism and alongside carbohydrate metabolism. They happen at the same time. So the major regulators for fatty acid metabolism are insulin, malonyl-CoA, also epinephrine, and ketone bodies are formed by excess fatty acid metabolism. If you have more than usual fatty acid metabolism, you'll be able to detect ketone bodies. All right, so this lecture was a little bit on the short side, but believe it or not, I think our remaining 2 lectures are both going to be even shorter. So I'll keep in touch with you over D2L for preparing for the 4th exam and the final exam. And please let me know if you have any questions. And have a great week.